हेलो एवरीवन, वेलकम टू NCERT ऑडियोबुक। आज हम क्लास 11 का चैप्टर 3 देखेंगे, प्लांट किंगडम। In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whittaker, 1969, wherein he suggested the 5 kingdom classification with Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with further classification with kingdom Plantae, popularly known as plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae through earlier classifications, placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm and angiosperm under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial system gave equal weightage to vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against these, natural classification systems developed, which were based on natural affinities among the organisms and considered not only the external feature but also internal features like ultrastructure, anatomy, embryology and phytochemistry. Such a classification for flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help to resolve difficulties in classifications. These becomes more important when there is no supporting fossils evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all over observable characteristics. Number of codes are assigned to all the characters and the data are then proceeds. In this way, each character is given equal importance and at the same time hundreds of characters can be important and at the same time considered. Cytotaxonomy, that is based on the cytological informations like chromosomes number, structure, behavior and chemotaxonomy, that uses the chemical constituent of plant to resolve confusion, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing simple thalloid, autotrophic and largely aquatic, both freshwater and marine organisms. They occur in variety of other habitats, moist stones, soils and wood. Some of them also occur in association with fungi, lichen and animals. Example on sloth bear. The form and size of the algae is highly variable, ranging from colonial form like volvax and filamentous form like eulothrix and spirogyra. A few of the marine forms such as kelps from massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being of zoospores. They are flagellated motile, and on germination gives rise to new plants. Sexual reproductions, flagellated and similar size as in Eulothrix, and non-flagellated, non-motile but similar size is an Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorrhenia is termed as unisogamous. Fusion between one large non-motile static female gamete and the smaller motile male gamete is termed oogamous. Example, Volvax and Fucus. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increases the level of dissolved oxygen in their immediate environment. They are of paramount oxygen in their immediate environment. They are the paramount importance of the primary producers of energy-rich compounds which form the basis of food cycles of all aquatic animals. Many species of Porphyra, Laminaria and Sagasum are among the 17 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrochlorides, water holding substance. Example, algin, brown algae, and carrageenan, red alga, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Glacidaria, is used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as a food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. 3.1.1 Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, collinear or filamentous. They are usually grass green due to the dominance of pigment chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospore produced in zoosporangia. The asexual reproduction shows the considerable variations in this type and formations of sex cells, and it may be isogamous, anisogamous and oogamous. Some commonly found green algae are Chlamydomonas, Volvax, Eulothrix, Spirogyra and Chara. 3.1.2 Phaeophyceae. The members of Phaeophyceae are brown algae are found primarily in marine habitat. They show great variations in size and forms. They range from simple branched filamentous forms, ectocarpus, to profusely branched forms as represented by carps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophyll pigment fucoxanthin present in them. Food is a source of complex carbohydrate which may be in the form of laminarin and mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by the gelatinous coating of algin. The protoplast contain In addition to plastids, a certain centrally located vacuole, protoplast, and nucleus. The plant body is usually attached to the substratum by the holdfast and has a stalk. The stipes and a leaf-like photosynthetic organ. The front, vegetative reproduction takes place by fragmentation. Asexual reproduction in most of the brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, and or eugamous. Union of gametes may take place in water or within the Euganimous species. The gametes are pyriform, pear-shaped, and bear two laterally attached flagella. The common forms of Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. 3.1.3 Rhodophyceae. The members of the Rhodophyceae are commonly called red algae because of predominance of the red pigment or phycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water areas. They occur in both well-lighted regions close to the surface of water and also at great depth in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as floridine starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentations. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is eugamous and accompanied by complex post-fertilization developments. The common member of Polysiphonia, Porphyra, Glacilaria, and Glidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in hills. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in the plant succession on bare rocks soil. The plant body of the bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, and leaves. They may possess root-like, leaf-like, and stem-like structure. The main plant body of the bryophyte is haploid. It produces gametes, hence it's called a gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated anthrozoids. The female sex organ is archegonium, is flask-shaped and produce a single egg. The anthrozoids are released into water where they come in contact with the archegonia, and anthrozoids fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives the nourishment from it. Some cells of the sporophyte undergo reduction division, meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are little economic importance, but some Mosses provide food for the herbivorous mammals, birds, and other animals. Species of sphagnum of moss provide a peat that have long been used as a fuel and as packing material for the transshipment of living materials because of their capacity to hold water. Mosses, along with the lichens, are the first organism to colonize rock, and hence they are great ecological importance. They decompose the rock, making the substrate suitable for the growth of the higher plants. Hence, mosses form dense mats of the soil. They reduce the impact of falling rain and prevent the soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the wood. The plant body of a liverwort is thalloid, example Markensia. The thallus is dorsiventrally and closely appraised to the substrate. The leafy members have a tiny leaf-like appendages in two rows of the stem-like structure. Asexual reproduction in liverworts takes place by fragmentations of thalli or by formation of specialized structure called gymi or gemma. Gymi are green, multicellular, asexual buds which developed in small receptacles called gemma cups located on the thalli. The gymi becomes detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into food, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free living gametophytes. 3.2.2 Mosses. The predominant stage of the life cycle of the mosses is the gametophyte, which consists of two stages. The first stage of the protonema stage, which developed directly from a spore. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears a sex organs. Vegetative reproduction in mosses is by fragmentations and budding in the secondary protonema. In sexual reproductions, the sex organs Antheridia and Archegonia are produced at the apex of the leafy shoot. After fertilization, the zygote develops into the sporophyte consisting of a food, seta, and capsule. The sporophyte mosses is more elaborated than that on liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Cephagum. 3.3 Pteridophytes. The pteridophytes include horsetail and ferns. Pteridophytes are used for medical purposes and soil blinders. They are also frequently grown in as ornamental. Evolutionary, they are the first terrestrial plant to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in the sandy soil condition. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well differentiated vascular tissues. The leaves in the pteridophyte are small, microphyll as a Selaginella, or large macrophyll as in fern. The sporophytes bear sporangia that are subtended by leaf-like appendages called Sporophylls. In some cases, sporophylls may form distinct compact structure called strobili or cones. Selaginella equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous small but multicellular free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow because of the specific restriction requirement and the need for the water for fertilization. The spread of living phaidophytes is limited and restricted to narrow geographical region. The gametophytes bear male and female sex organs called antheridia and archegonia respectively. Water is required for the transfer of anthrozoids. The male gametes released from antheridia to the mouth of archegonia. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote therefore after produces a multicellular well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kind. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of the spores. Macro, large, and micro, small spores are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophyte, respectively. The female gametophyte in these plants are retained on the parent sporophyte for variable periods. The development of the zygote into young embryos takes place within the female gametophytes. This event is an precursor to a seed habit, considered an important step of evolution. These takes place within the female gametophyte. This event is the precursors to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Coelopsida, Coeloptum, Lycopsida, Selaginella, and Lycopodium, Ctenopsida, Equisetum, and Pteridopsida, Dryopteris, Pteris, and Adiantum. 3.4 Gymnosperm. The gymnosperm, gymno-naked spermacid, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seed that develop post-fertilization are not covered, that is, are naked. Gymnosperm include medium-sized trees or tall trees and shrubs. One of the gymnosperm, the giant redwood tree, Squinia sequoia, is one of the tallest tree species. The roots are generally taproot. Roots in some genera have a fungal association in the form of mycorrhiza, pinus, while in some other cycas, small specialized root called choroid roots are associated with entomophyxine cyanobacteria. The stem are unbranched cycas or branched pinus or kidrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extreme of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spores are produced within sporangia that are borne on sporophyll, which are arranged spirally along an axis to form a lax or compact strobili or cones. The strobili bearing microsporophyll and macrosporangia are called microsporangiate or male strobili. The microspore develop into male gametophytic generation, which is highly reduced and is confined to only a limited number of the cells. This reduced gametophyte is called A pollen grain. The development of pollen grains takes place within the microsporangia. The cone-bearing megasporophyllae with ovules or megasporangia are called megasporangiate or female strobili. The male or female cones of strobili may be born on a same tree, Pinus. However, in Cycas, male cones and megasporophyllae are born on different trees. The megaspore mother cell is differentiated from one cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are born on megasporophyllae which may be clustered to form the female cones. The megaspore mother cell divides meiotically to form 4 megaspore. One of the megaspore enclosed within the megasporangium develops into the multicellular female gametophyte that bears 2 or more archegonia and female sex organ. The multicellular female gametophyte is also retained with the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperm, the male and female gametophyte do not have an independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in the air current and comes in contact with the opening of the ovules born on megasporophyllae. The pollen tube carrying the male gametes grown towards the archegonia in the ovules and discharge their content near to the mouth of the archegonia. Following fertilization, zygote develops into the embryo and the ovules into seeds. These seeds are not covered. 3.5 Angiosperm. Unlike the gymnosperm, where the ovules are naked in the angiosperm of flowering plants, the pollen grains and ovules are developed in specialized structure called flower. In angiosperm, the seeds are enclosed in fruits. The angiosperm are an exponentially large group of the plant occurring in wide range of habitats. They ranges in size from smallest Wolfia to tall tree of the eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into 2 classes, the dicotyledon and the monocotyledons. The dicotyledons are characterized by the seeds having 2 cotyledon, reticulate veneation in leaves and tetramerous or pentamerous flower, that is having 4 or 5 members in each floral whorls. In monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel veneation in leaves and trimerous flower having 3 members of each flower whorls. The male sex organ in the flower is the stamen. Each stamen consists of a slender filament with the anther at tip. Within the anthers, the pollen's mother cells divide by meiosis to produce microspore, which matures into pollen grains. The female sex organ in the flower is pistil. Pistil consists of solan. Ovary are present. Generally, each ovule has the megaspore mother cell that undergo meiosis to form 4 haploid megaspore. 3 of them degenerate and 1 divided to form the embryo sac. Each embryo sac has a 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce the diploid secondary nucleus. Pollen grains after dispersal from the anthers are carried by wind or various other agencies to the stigma of the pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grow through a tissue of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with egg cell syngamete to form zygote. The other male gamete fuses with the diploid secondary nucleus to produce the diploid primary endosperm nucleus (PEN). Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization. an event unique to angiosperm. The zygote develops into an embryo with one of the two cotyledons, and PEN develops into endosperm, which provide nourishment to the developing embryo. The synergids of antipodal degenerates after fertilization. During these events, the ovules developed into seeds and the ovaries developed into fruit. The life cycle of an angiosperm is shown in figure 3.6. Let's see the figure 3.6, where the sporophytic generation and gametophytic generation of angiosperm given here. 3.6 Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant bodies produce gametes by mitosis. This plant body represents the gametophyte. Following fertilization, the zygote are also divided by mitosis to produce the diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form the haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between the gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following pattern. 1. Sporophytic generation is represented only by the one-cell zygote. There are no free-living sporophyte. Meiosis of this zygote results in the formation of haploid spores. The haploid spores divide mitotically to form the gametophyte and dominant photosynthetic phase in such plants, the free-living gametophyte. This kind of the life cycle is termed as haplontic. Many algae such as Volvax, Spirogyra and some species of Chlamydomonas represent this pattern. 2. On the other extreme is the type within the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single or few-celled haploid gametophyte. This kind of the life cycle is termed as a diplontic. An alga fucus species represent this pattern. In addition, all seeds-bearing plants like gymnosperm and angiosperm follow this pattern with some variations, wherein the gametophytic phase is few to multicelled. Third, bryophytes and pteridophytes distinctly exhibit the intermediate condition, haplodiplontic. Both the phases of multicellular, however, they differ in their dominant phases. A dominant and independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by dominant independent photosynthetic vascular plant body. It alternates with multicellular sporophytic autotropic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most of the algal genera are haplontic, some of them such as Ectocarpus, Polysiphonium, Calps are haplodiplontic. Fucus and alga is diplontic. Here the third lesson, plant kingdom, is over. Thank you.